Legionella pneumophilia modulates mitochondrial dynamics to trigger metabolic repurposing of infected macrophages. A video by Anna Rappaport, Desiree Dean, Jasmine Lucio, and Will Flanagan. Macrophages are large white blood cells that play an important role in the immune system. They use the process of phagocytosis to recognize, engulf, and digest any microscopic foreign bodies. The metabolisms of macrophages are refined by mitochondria. Mitochondria are highly dynamic organelles in which respiration and energy production occur. Additionally, mitochondria have functions in the regulation of cell death and innate immunity. Bacteria have evolved complex mechanisms to target mitochondria and exploit the key roles they play in the cell. Legionella pneumophilia is a gram-negative intracellular bacterium and the agent of Legionnaire's disease. Symptoms include shortness of breath and fever. L. pneumophilia has a type 4 secretion system to disperse the bacterial proteins that hijack host functions. The T4SS targets host organelles such as the nucleus and endoplasmic reticulum. This study focused on L. pneumophilia-induced mitochondrial fragmentation. During the study, the T4SS effector responsible for fragmentation was identified and named mitochondrial fragmentation factor. The researchers examined the relationship between L. pneumophilia and the oxygen consumption rates in mitochondria. To their surprise, researchers found an upregulation of oxygen consumption within the first 2 hours of infection in both wild-type and mutant L. pneumophilia infected cells, suggesting that the regulation of cellular respiration was T4SS independent. However, oxygen consumption rates began falling 2 hours post-infection in the macrophages infected with wild-type L. pneumophilia, meaning that regulation of cellular respiration after 2 hours is T4SS dependent. The researchers attributed this decline to T4SS dependent morphological changes in mitochondria due to the increased activity of DNM1L, a protein triggered by MITF that causes mitochondrial fragmentation. To further support DNM1L's role in mitochondrial fragmentation, the researchers used interfering RNA to silence expression of DNM1L. Upon doing so, the researchers found that mitochondrial fragmentation decreased from 23.26% to 9.74% in the presence of iRNA. Despite this, the researchers wanted more evidence of a T4SS-dependent relationship in DNM1L's activity level. Upon infecting cells with mutant and wild-type L. pneumophilia, researchers found significant differences in DNM1L levels between wild-type infections and mutant infections. A secondary function of DNM1L is triggering cell death, but the researchers found that this is suppressed in L. pneumophilia infections. Instead, the decrease in respiration caused by DNM1L results in the upregulation of glycolysis. The increased glycolysis is actually beneficial for L. pneumophilia as its byproducts can be used for cell growth and replication. This works much like the Warburg effect, which is common in cancer cells. In summary, L. pneumophilia causes a severe type of pneumonia. The T4SS effector MITF induces DNM1L dependent mitochondrial fragmentation. L. pneumophilia creates a Warburg-like metabolism in the host cell that favors bacterial replication and suppresses cell death.